Assalamu alaikum. My name is Farhan Farabi, and our project title is Breast Cancer Detection Using Image Processing Techniques. These are the names of our group members and their contribution. So, our problem statement is manual diagnosis of histogram images or mammogram images tends to be much time consuming and sometimes leads to wrong assumptions due to human error. And slower diagnosis leads to faster cancer spread and eventually death. To solve this problem, traditional machine learning was used for processing images to identify whether the image is cancer or not cancer. But these traditional image learning techniques, such as uh, support vector, are very slow and sometimes with less much accuracy. And and researchers found that previously, using machine learning techniques in traditional machine learning techniques, we could identify whether the image is benign cancer or malignant cancer. So it turned out to be only binary classes. But recent studies show that classification of cancer images are classified as multi-class problems. So keeping these problems in mind, we propose that we use deep learning techniques such as CNN, convoluted neural network architectures, to solve this multi-class problem. Uh, what CNN does is CNN has inputs many hidden layers. Hidden layers, that means it has more than 150, 50, sometimes 60 layers. These layers convolute these layers take the input image and process it by reducing the size of the image and taking more kernel inputs to give an output of a much specified output. So using our knowledge of the course, we built a classifier using CNN architecture to accurately identify benign and malignant cancer images. So training classifiers using CNN architecture solves the problem of diagnosis and late diagnosis and outperforms traditional machine learning techniques. So here I will describe the rest here. So this is our methodology. So first we input the histology image, then we through some pre-data processing we resize that image to 224 pixels. Then in that image we find out the breast area, and after some data augmentation we process that data, and then we send that data to the classifier. So here we use a pre-trained classifier because ResNet classifier found that there was more than 2 million parameters. So we only trained about 2,400 parameters. So here we use ResNet, more specifically ResNet 50. 50 means 50 layers, V2. So after training that module, we could identify whether, and after training, because we took a data set, then we split that data set into trained data set and test data set. So after training that data set, we tested that with some cancer images and found whether it was benign and malignant, and we were successful. So here is the process of CNN. First let me explain the ResNet. So what ResNet does is it uses a skip connection. So here identity function X, we use it at input image, and then the identity function is used in the last stage. So here weight layers, so these are some convolution layer, and these are the fx, our function loss function. We add the identity X with the fx such so that we can make the fx 0. By doing so, we make it output is equal to input. So doing this, what it does is it increases the accuracy. So this is the residual learning block, mostly ResNet. So here is the process. So it takes the image, then this is the first layer. So what it does is it reduces the image size. So 7 by 7 is the convolution layer size, and 64 means the kernel. So what it means that it will have 64 sets of filters. So then it uses another layer for max pool. So we have two types of pool, max pool and average pool. So here we are using max pool. So, and also we use a stride we use to what stride does is it downsamples. That means we by increasing the stride, we actually reduce the size of the image. So after doing that, these are the layers. So one by one, first we do one by one convolution layer with 64 kernels, then again one by one convolution 64 kernels, then one by one convolution with 256 kernels. This layer is done 3 times, and with every one or two layer we do a skip connection. So, and here after this layer, after this has been done 3 times, here the input identity X is different, and this is passed in another layer, which we have 1 by 1 convolution with 128 kernels. So what it does is, a convolution layer is the main building block of a CNN. It contains a set of filters or kernels like 64 or 255 or 128, and parameters of which are to be learned throughout the training. The size of the filters is usually smaller than the actual image. That is, the 7 by 7 layer, we take a small part, and then we actually take within that range we actually take the max value. Since we are using max pool, we are taking the max value from that part and processing into the next layer. So for the convolution, the filters leads across the height and width of the image and the dot product between every element of the filter, and the input is calculated at every spatial position. So and after doing this, we create an activation map. So activation map is generated by repeating this process for every element of the input image. So yeah, we do this for every element, so creating a 3D volume of that image. The output volume of the convolution layer is generated by stacking the activation map of every filter along the depth dimension. So every component of the activation map can be thought to be an output of a neuron. So after doing this, we take the average pool. So that means after convoluting, so here we can see, so the 3 times, this is done 3 times, this is done 4 times, this is done 6 times, and this is done 3 times. And after calculating all the layers, we find that there are 50 layers. So that is why we have used ResNet 50 v2. So we get the output volume. So output volume, after getting the output volume from here, we can see that the image is passed through the ResNet module. Then after we get the 3D output volume, we do a global average pooling, and doing so we flatten that 3D volume into a 1D vector and do the similar computation like we do in an MLP. Then after that we do the classification. There are 4 classes here, but we have 2 classes, either benign or malignant. So doing this, we will get an output of whether the image is benign cancer or malignant cancer. So here I stated that this fx is our loss function calculation. So here we used binary cross entropy as our loss function. Binary cross entropy compares each of the predicted probabilities to actual class output, which can be either 0 or 1. So we have put 0 as benign and 1 as malignant. If the calculate the score that penalizes the probabilities based on the distance from the expected value. That means we actually calculate how far or how close the loss function is actually from the actual value. So, and we also used an optimizer of Adam. So Adam optimizer Adam optimizer involves a combination of 2 radiant descent methodologies. Momentum. This algorithm is used to calculate the gradient descent algorithm by taking into consideration the exponentially weighted average of the gradient. Usually, using average makes the algorithm converge towards the minima as a faster pace. So we know there are local minima and global minima. So after using Adam optimizer, we will get to the global minima much quicker. So this is our main process of building a classifier. So after that, we have tested it. So we found the following results. The results stated that the training accuracy was 0.9290, the training loss was 0.2087, and the validation accuracy was 0.9130, and the validation loss was 0.2764. So here is a visual representation of our training, and we could see that our validation accuracy and total accuracy is closer to 1, and our losses of validation loss and the loss is closer to 0.2. That means our model is performing very well, and we can see that ResNet 50V is giving a better accuracy because ResNet is using a skip connection. Researchers found that having deeper layers sometimes tends to be much less performing than shallow models because of having too many parameters in the end phase. So using ResNet what it does is it actually keeps the uh, it keeps in check the identity function or the input value. So doing this, what it does is it actually increases the accuracy amount, and we get a more better model that can predict whether if it is cancer or not. So in conclusion, we can see that deep learning models can, with using deep learning models such as ResNet or GoogleNet, we can actually create a classifier which is a much better predictor for cancer from images than traditional machine learning algorithms. So, and from the content that we have learned in the class, we were successfully able to create a classifier, and and keeping in mind all the researches and all the procedures, we found a much better accuracy in CNN models. So now I'm going to explain the our code module.
Assalamu alaikum. In our project, first the breast cancer classification project. First, we have collected some data sets from Google here. In these data sets here, we have two folders with 0 and 1. So 0 is 10 for the images with no breast cancer images, and we can say there are a lot of data, lot of images here, and also in the folder 1 we have the images that has the images of breast cancer. So there are a lot of files here, and it takes a lot of time to import them in Google in the Colab. So we have added our Google Drive here, and we will be using this Google all the files the our all the files in this Google Drive link here, and we have added the path of the drive link here. And after that we have implemented our necessary Python libraries. So we can see there are NumPy, TensorFlow for machine learning, and Pandas, Matplotlib, and also we are using Keras, so that to use neural networkings in these in here, and using also CV2 for some OpenCV file. And after that we have added the data here, and also the data sets of no breast cancers and yes breast cancers. So in the second part we are using these to see how many files are here, how many labels are here. And also in here what we have been doing is like we have been using a data frame here in this file. So like what we have got from the file paths and labels, we are just making a data path here. Like here we can see that in the row there are a column of the file path, and for every file path there is our labels are set up here. And now here we have made here for our some visualization. This one actually was created for only visualization. There are some random images for the visualization purpose only. And now here we are training our data sets here. So first there will be two parts, like we will be training the data set. So there will be train and test, and after that the train part will be validated. So there is also shape of the validation part. So and we are also printing the shape of the data sets here. And now in this part what I'm doing is some image data generation, which simply means I'll be doing here some augmentation here. And now here we have created a training set, validation set, and test set. In the training set we are using the data frame of train new, which is in here, this one. In the here we are also giving the X column for file path and Y column for labels here. Gina. Also here we have also has the target size. The target size means basically the image size, which image size we are going to consider in here. We can see we are using here the 220 to 224, which is working fine in this project here. Okay, here we are doing the some transfer learning. Here we are using Keras.application here, and here we are using the ResNet 50 model here. We are giving the input shape of the image is 224 224. Okay, in here we will use a ResNet in our as our base model. So base model here, and after that we will be using making the base model freeze here, and we will be using some here we are after that we are using three layers like global average pool layer, dropout layer, and finally the output layer is here. And we can see that we are using only one set of output here, like it's a binary classification problem, so either we will be using yes or we will be using no. And here we have used some functions like callback functions. So the what the callback function is basically do is like it will save our model in sometimes for example, if we're working with 100 epochs, so if we get like good accuracy till 50 epochs, then it will save it here and then will function again. So this is what it basically does. And also we are using a model.compile here. So in the loss we are using the binary cross entropy, and the optimizer we are using here is Adam. And we can see that we have after that we have used the model.fit here. We fitted our model with the epochs of 100 epochs, and it then calculated the loss and the accuracy functions here. And here we have saved our model with a model.h5 here. This is also saved in our Google Drive, like here it is saved. So after that we have evaluated the model with the datagram, and here like loss, accuracy, these have been saved here. How was our accuracy here. And here we have just like testing the data model with some pictures. And first of all we have loaded our model here, and we have used also the numpy function here that we will be needing to size the pictures here. So as we have seen that we have given a image here to test our function, and there was causing a problem with the segment function. So we have added a condition that if the value gives we will be needing the value with 0 and 1, which will be yes or no. So if the value is greater than this threshold value 0.5, it will show yes, and if the value is less than it, then it will show no. So this image is also uploaded in our Google, in my Google Drive link. So this is how we have been testing the set. So as we have seen there, we have taken non-breast cancer images, and our model was able to show that it was a non-cancer. And if we take an image from the cancer images here, so let's see if it's able to check it correctly or not. So to check it, we will be taking these files, and now as we can see that it has successfully checked the dataset of the yes breast cancer.